Namaskar. Today we'll study the ribosome and protein synthesis. So ribosome has two subunits. One is the larger subunit and the other is the smaller subunit. Okay. And in eukaryotes it is 60S and 40S, and this complex is called 80S ribosomal complex. But in prokaryotes like your bacteria, it is the larger subunit is 50S and the smaller subunit is 30S. So 50S and 30S combined together, it is 70S ribosomal complex. So the messenger RNA binds to the smaller subunit and transfer RNA binds to the larger subunit. So mRNA binds to your 30S in bacteria and tRNA binds to your 50S in bacteria. And the function of ribosome is protein synthesis. So how this protein synthesis occurs, we can see each messenger RNA has a base sequence that we have seen in earlier class, that messenger RNA will have some base sequence, and that base sequence three consecutive base taken together is called the codon or genetic code. So messenger RNA has the codon, that is, it is called messenger RNA because it has got the message which type of protein has to be synthesized. So the DNA is the carrier of the genetic information. So from DNA, mRNA, messenger RNA is produced by the process of transcription. Then this messenger RNA has a codon, and to each codon there is anticodon, and you can see this tRNA. tRNA has anticodon, so three bases here complemented to that of the mRNA, and this anticodon is linked to one amino acid. So in tRNA, at one end we have amino acid and the other end we have anticodon in triplet, and that anticodon is complementary to that of the codon in the messenger RNA. So once the codon of messenger RNA is recognized by the anticodon of transfer RNA, then that amino acid will be supplied, so that one after another amino acids will come and they will join, and we will have the protein or peptide synthesis. Okay. So first messenger RNA will bind to the smaller subunit, say 30S subunit, with initiation factors. That complex will be formed. Then 50S subunit and transfer RNA. So transfer RNA will bind to the 50S subunit, and the 70S initiation complex will be there. And the anticodon of transfer RNA will recognize the codon present in the messenger RNA, and it will supply the required amino acid. Required amino acid will come one after another, and the protein will be synthesized. So how this occurs? So we know there are four bases. So in RNA we have A, U, C, G. Four bases are there. So four bases will produce 4 to the power 3. So 4 to the power 3 is your 64. So there will be 64 codons. You can see the first base U, second base U. So U, third base U. Here we have written UCAG, UCAG, UCAG like that. So first base, second base, third base, U. First base, second base, third base, UCU. And accordingly UUC, UUA, UUG. And if we prepare this list of codons, then we will get 64 codon. And out of 64 codon, we have actually total 20 amino acids. 20 amino acids, we can divide them into essential and non-essential. The essential means we have to take them in our food. We cannot synthesize them. So they are essential amino acids. And the non-essential amino acids which we can synthesize in our body. No need to take them if through food. So then again we can call some of them as conditionally essential, because synthesis is limited under special pathophysiological conditions, for example premature babies, and dispensable, they are synthesized in sufficient quantity. So all these amino acids, they are 20 amino acids, and these amino acids, all these 20 amino acids are represented by 64 codon. Out of that 4 codon, 3 stop codons are there: UAA, UAG, UGA. These 3 are stop codons, and AUG is the start codon, which is always methionine. So methionine codes for AUG, and the rest 19, you can see here, leucine has 6 codon: UUA, UUG, CUU, CUC, CUA, CUG. Likewise, certain amino acids have 4 codon: GUU, GUC, GUA, GUG for valine, and similarly some have 2, some have 1 codon. Okay, and total 20 amino acids with 64 codon, we can represent them. So when one codon, suppose the CUC is the codon in the messenger RNA, then the transfer RNA will have anticodon C for G, U for A, C for G. So the anticodon will be G, A, G. So the transfer RNA with anticodon GAG will recognize this codon, and which amino acid will be there at the other end of transfer RNA? Leucine will be the amino acid. So accordingly, if we see this example, so we have DNA, and DNA, one strand will be sense strand and the other strand will be the antisense strand, and the antisense strand will act as the template. By the action of DNA dependent RNA polymerase, the messenger RNA will be produced, and the messenger RNA will have complementary bases to that of your antisense strand and similar bases to that of your sense strand, because your sense strand and antisense strand are complementary to each other. Then the only difference will be the messenger RNA has same base sequence as that of your sense strand, except U for T in the sense strand. Then suppose this is the first codon AUG, and AUG codes for methionine, which is the start codon, and so the transfer RNA at one end it will have methionine as the amino acid, and the other end the anticodon UAC, so it will recognize. So first amino acid provided will be methionine. Then similarly the second one GGG is the codon, so CCC will be the anticodon with transfer RNA, and CCC this is representing proline. So GGG is the codon and CCC is the anticodon. So GGG will be the codon and representing glycine. So the transfer RNA will have glycine as the amino acid, and in this way all these codon will have codon representing one amino acid will recognize this transfer RNA with anticodon and respective amino acid. Then all these amino acids will bind until the stop codon comes in the mRNA. If the stop codon, there are 3 stop codons, UAA, UAG and UGA. So once the stop codon comes, the protein synthesis will stop, and we will get the peptide. So in this way we can synthesize this protein synthesis occurs. So if we want, suppose in bacteria, if we want to inhibit protein synthesis, then we have to selectively target 30S subunit, so that the 30S mRNA complex formation will not be there, or we can target this 50S subunit so that 50S tRNA binding will not be there. So in this way we can inhibit protein synthesis in bacteria. So the drugs like your tetracycline aminoglycoside antibiotics, they inhibit 30th subunit of ribosome, and drugs like your macrolide antibiotics like erythromycin, azithromycin, they inhibit 50th subunit of ribosome. So this is all about protein synthesis. Thank you.